In today's lecture, we'll delve into the fascinating world of bacterial genetics and explore one of the primary mechanisms by which bacteria exchange genetic material, bacterial conjugation. Bacterial conjugation is a process of horizontal gene transfer, where genetic material, typically in the form of plasmids, is transferred from a donor bacterium to a recipient bacterium through direct cell-to-cell contact. This process plays a crucial role in the rapid spread of antibiotic resistance and virulence factors among bacterial populations. Let's meet the key players involved in this process. Donor cell F plus cell. This bacterium carries an F plasmid, which contains genes necessary for conjugation, including the genes for pilus formation. Recipient cell F minus cell. This bacterium lacks the F plasmid and is thus capable of receiving genetic material. Pilus, a hair-like appendage on the donor cell that extends and attaches to the recipient cell, forming a conjugation bridge. Now, let's walk through the steps of conjugation. Pilus formation and attachment. The donor cell extends a pilus, which attaches to the recipient cell. This forms a conjugation bridge between the two cells. Transfer of F plasmid. A single-stranded copy of the F plasmid is transferred from the donor cell to the recipient cell through the conjugation bridge. This transfer is unidirectional, moving from the donor to the recipient. Replication of F plasmid. Once the F plasmid enters the recipient cell, it replicates to form a double-stranded copy, making the recipient cell an F plus cell. Separation of cells. The conjugation bridge breaks, and the two cells separate, each now possessing an F plasmid. Want to learn more about bacterial conjugation and other fascinating topics in biology? Visit our website, biologynotesonline.com, for free study notes, quizzes, flashcards, and more resources to deepen your understanding.